Okay, I think we're kind of at capacity, so we'll get started. And welcome to our webinar around challenges and solutions for managing clinical study data or clinical trial data. I'm Hannah Brackey. I'm a product manager here at LabKey. I've been here for about 6 years, but prior to that, I worked in a translational lab, collecting, managing, analyzing samples for our pediatric CAR T-cell research at an institution here in Seattle. I've used a CDMS before in terms of trying to wrangle some of this data. And it's very near and dear to my heart to try and talk to you guys about this today and help you figure out a solution that's going to work for you. So a couple things about LabKey as a company. We really pride ourselves on being more than a software company, and we really have a long history in working and collaborating with scientific organizations and labs to build sustainable, scalable solutions for them in their scientific data management. We started over 20 years ago now as a spinoff of the Fred Hutch here in Seattle. And we have a lot of expertise in managing data, but specifically in managing data that is science-oriented. And that really is going to shine today as we're talking about our CDMS, this clinical data management system. We also have a very unique culture here at LabKey where the service team and the organization as a whole is very, very invested in our clients' success. And we have a high level of support that we provide people, and understanding that oftentimes you're coming to us without a system. And so this is your first system, and being able to provide that advice and guidance kind of as we're going through. So just nice to talk about our experience, so you can understand where we're coming from when we're talking about sort of the importance of that CDMS. We also see a lot of our clients sort of struggling with the massive amount of data that comes out of clinical trials or clinical studies. And so using that experience and really paying it forward for new groups that are joining us as well. So hopefully this is not that much of a surprise to anyone who's here, but there are a lot of components that are surrounding a study. And usually I like to categorize these very, very generally as sort of the data inputs. So the collection piece of what's happening throughout a study. So EDC data is a very common one, or CRF data. So effectively, if you're using an electronic data capture system like REDCap or metadata or some other system, or if you're collecting those on paper, you have something happening around those CRFs. There are often samples that are being collected, potentially used and tested on, or also stored in a biorepository or something similar. There can be lab data associated. So if there is either clinical lab data with sort of standard test results, or even more extraneous translational lab data, such as what we were doing of running flow cytometry and gene expression and things like that, you tend to have a lot of different formats of this data. EHR or EMR data, so sort of the medical data that's associated with these patients or subjects that are part of the trial. And then potentially even external databases. So if you have a number of collaborators or a large enough consortium, or potentially enough legacy systems, there might be other databases that have vital information around the trial that you want to use for your downstream analysis or things like that. And then there's really the output component, which is kind of everything here on the other side of the screen. So things much more related to the day-to-day study management. So interested in enrollment. What does that data quality look like? Have there been reviews? Are we missing data? Do we need to understand what's happened there, as well as sort of understanding that overall study progress? There's that reporting piece. I have kind of listed a couple of common reports. If you have reports that you're generating for your trials, please add them into the chat. And I'd love to try and talk about them when we're in our demo today. But these reports are often very safety related. So adverse event or severe adverse event reporting. Oftentimes these reports have to come very quickly, and having a system to help generate that is very useful. Sort of standard oversight committees like the Data Monitoring Committee or the Data Safety Monitoring Board will often require sort of collation and cleaning of data for these reporting bodies. And then finally, of course, the analysis, right? So there's going to be the statistical analysis to help sort of measure efficacy and safety and things like that, but then also sort of the ancillary pieces of analyses that really go around sort of data sharing or potentially sort of publishing results. Nowadays, we're all talking about sort of predictive analysis. So having clean data to put into sort of those predictive models can be very useful as well. And we have this sort of squiggly line between these inputs and outputs that oftentimes folks are struggling with when they're running trials, and having a hard time kind of getting these outputs in an efficient and clean and reliable way. And so that's where kind of the concept of a CDMS comes in. So when we talk about what these challenges of that squiggly line are, I have a couple ideas in mind of what we've seen clients experience, what I've experienced. If you have challenges here, I'd love to sort of see you add them to the chat. So I'll kind of start out by just saying sort of like siloed data is one, right? We just talked about all of these different inputs. It's very likely that all of these are being collected in different systems by different people, potentially across sites. And so to have all of that data under one roof can be very useful. Does anybody feel the desire to share? It looks like not, which is totally fine today. So I'll just kind of jump into the ones that I think. So we talked about sort of that siloed data. Having data in these different systems, but then also really in these different formats. So when you're talking about EDC data, if you're using REDCap or others, there's a CDISC format, a very standardized format that these come in that are harder to deal with kind of from a human perspective. You'll want to make sure that you're able to sort of reconcile that data across all of these different silos in order to produce those outputs, reports that we were mentioning in the previous slide. There's a data cleaning and analysis and reporting challenge as well. So oftentimes, especially when we're collecting a lot of information for trials, without sort of robust systems, there tends to have to be a lot of data cleaning that comes with that, and making sure that, you know, values that are being entered are within range and accurate and of the right format. Making sure that we're also validating that data. And so is that are those sort of valid responses for the question that we're asking folks to enter? Are those test results sort of within range of what we anticipate? And then ultimately, you know, the quality control component or the QC process that comes into play to make sure that all of these things are as accurate and reliable as possible. Making sure that we're also aligning these different data types. So I remember back in my previous life, you know, comparing multiple spreadsheets on one screen and trying to get them and understand, you know, what time point across all of these different spreadsheets? Can we sort of gather and collate this data for maybe an adverse event report or something else? So that often is very time consuming. It can be even more time consuming when that first bullet point has its challenges. Making sure that we understand sort of that participant information and really being able to view data across time points. So what is sort of the common thing that happens at visit 3 versus visit 10 really tends to take a lot of effort, especially if you have sort of data cleanliness or even sort of this siloed data problem. And then ultimately the reproducibility, right? So we want to know where a piece of data came from. More often than not, you get to publication and you want to do something, you have maybe a review, and you need to trace back, what is it that you're reporting, and where did that come from, and how can we continue to make that sort of robust and reproducible. And so having a system that allows you to make that connection can really help in that downstream reproducibility. And then, of course, we're talking about human data. So making sure that things are secure and shared appropriately. It's oftentimes, you're not doing trials in a vacuum. You are working with collaborators. You want people to have the appropriate access to data, and of course, the appropriate controls so they don't access incorrect data that they don't they shouldn't be seeing. So having that proper access control is something that oftentimes it is a struggle without a system. Making sure again that your data is clean enough and reproducible enough to have these publish ready reports or charts, making sure they also get de-identified in the proper way as well. And then, of course, providing audit trails. So understanding when data has changed and why it's changed, and maintaining kind of that regulatory compliance piece that's so vital to running trials. There's other challenges here that folks on the call have encountered. Feel free to sort of add that to the chat, and we'll take a look at it as well. So as I've kind of alluded to, right, the squiggly lines, these challenges of getting the inputs to the outputs can really be helped very greatly by a CDMS or a clinical data management system. And centralizing all of these different pieces of siloed, varying formatted data into a centralized location allows us to ease the path for these outputs that we're looking for, either on sort of the ongoing study management piece or more of the downstream reporting and analyses that we're really interested in. CDMS in general, we often get this question is, what is different of a CDMS versus something like a CTMS, which is a clinical trial management system? Clinical trial management systems tend to be more responsible for the scheduling and the organization of the trial, kind of that project management piece of it. And CDMS can actually pull in information from a CTMS to help centralize and collate this. So again, you're looking at centralized and common dashboards across maybe a CTMS versus an EDC versus sort of laboratory data that folks are looking at. So really getting that comprehensive study overview is the power of a CDMS in general, and of course of LabKey CDMS as well. So if it wasn't already obvious to talk about kind of the benefits of a CDMS, there is certainly that improved data quality piece of it. So being able to provide enforcement checks on data entry, and then ultimately tracking sort of the quality control or the process involved to validate data is something a CDMS should bring to the table. really helping to minimize manual data entry as well. So if you are able to pull data in from different systems, somebody's not looking at a screen at the EDC and typing it in somewhere else, there's ways to programmatically centralize and pull this data together. And then also providing kind of a common data entry method for either remote sites or potentially just the multitude of personnel that might be adding information about this trial. And so having that centralized location again is really nice to create sort of that source of truth. Improving the data structure. So one of the things that's often overlooked is sort of better data structure provides better data in the end. And so if you can find a system that is designed to track studies and participants and visits and all of the information sort of associated with those, that's going to really help smooth out that analysis and reporting downstream. So you're not always maybe having to either manually go through Excel files and do things with, varying risks of what Excel does, but really sort of go in and with a few clicks, create a report that's going to be reliable and useful later. And of course, sort of having that improved data structure as people scale, avoiding those issues that happen with scalability as well. You have a system in place that basically sets you up for success in sort of a long-term ongoing trial or expansion to other studies as well. This is actually a very sort of famous story internally at LabKey. When I first saw LabKey maybe 14 years ago when I was in this translational lab, I thought a CDMS is too powerful for us. We have one study and one patient right now. This feels really, really heavy-handed for us to take on. And my boss very astutely in the moment said, but we're not going to stay with one patient and one trial. And I think now that group is up to 14 trials and sort of hundreds of thousands of patients. So having kind of that scalability very early on really it can be very helpful. Enhancing that collaboration. So I mentioned the single source of truth, having kind of that single access point for teams, I think especially as you have remote sites or other sites, being able to easily communicate and provide portals for those sites to view data that they should have access to really is very helpful. Also being able to provide sort of more efficient coordination and workflow. So with a centralized location, a single source of truth, all of that comes very naturally. And I see someone's raised your hand. So please feel free to sort of add any questions in the chat too. And I'll try and just keep a lookout for that and answer them as I see them. And then lastly here, a better foundation for regulatory compliance. And this can be something that is it's very, very important to maintain this compliance. Using systems that help with this really, really help. So providing systems that have rigorous auditing and really being able to troubleshoot or track or understand how some data got to its state is very valuable when you're running trials, especially sort of at that scale. So I think it's time we kind of look at a CDMS in action. So we've got a completely sort of fabricated demo to share with you that's comparing ARV treatment regimens in participants that are infected with HIV. In this fabricated environment, we have data that's being collected from a REDCap EDC. We also have other data that's coming from our in-house virology lab that we want to pull in and understand viral loads and things like that. There's also a biorepository that has samples that we may be using for sort of current analyses or storing for extended analyses later. And then also data from, in this case, sort of collaborators who are generating gene expression data for us. So that would be kind of that external database that we're linking to. And so here's kind of the dashboard of my CDMS. This is really a dashboard that is study focused. So I'm looking at this individual study around HIV progression. You can see I've kind of flipped the diagram from the slides in sort of this 90 degrees, where we have all of the input data. So our EDC data and our lab data, our sample data, and this external database data is kind of being listed here. All of these are links into those tables that have been pulled in from these other sources. And then I have kind of the outputs that we're interested in. And so the study management component and our reports that we may need and the analysis that we're doing. So having kind of this centralized dashboard allows me to click through very quickly if I'm looking to generate something or want to understand what needs to be tested. The CDMS in general has this concept of participants built in. So essentially, when I provide a participant notion, the CDMS is very good at understanding how to parse that information and say, okay, we've got, these 12 participants as part of this study. We also have a cohort notion. So I'm able to look at my participants based on what cohort they've been assigned. So we can do sort of cross cohort comparisons or collation as we need to. And then I also have created my own group around sort of HIV status, again, to be able to make those easy comparisons between participants within my study. All of that data that I had kind of nicely displayed on the dashboard is also available in this data and reports tab. And this is all very customizable. So I pulled in data from my EDC that was called demographics and enrollment and adverse events. You may have many others, CRF forms that are associated with that. But all of these are completely customizable by an administrator and perfectly sort of aligned with the data capture that I have in this fabricated study, of course. So if I go back kind of to that dashboard and take a look at some of these things, so I'm able to look at my EDC data, for example, and understand what is that data that has been pulled in from the REDCap EDC. And we're also applying some quality control measures here within the CDMS. And so at this stage, I'm able to see what I'm not able to sort of what hasn't passed review versus what still needs to be reviewed. And the other piece that I wanted to share here is that this is a really great example of those access controls. So right now, I'm not a person that has the access or privileges to look at a participant's birth date because that might be considered sort of PHI. And so we have access controls both at the full table level. So all of the tables that we're seeing are tables that I've been explicitly granted access to, but also at the column level. So if there's data that is sensitive or PII or PHI, we're able to apply that and then apply the appropriate permission to those users as well. So if I go back to the study dashboard, most of these other tables are going to look very similar. It's just going to be sort of a tabular list of all this data. That's actually wonderful because most of these were not very tabular coming from other systems. So things like our PCR data had a lot of sort of transformation that needed to get applied that was programmatic when we entered it in. The same with that EDC data and sample data. So if we go to the output, excuse me, which is the thing we probably all most care about, I can now sort of click in and look at these individual things. So if I look at my enrollment report, I'm able to understand, oh, we've got somebody that hasn't consented. Is it because they've deconsented, or do we need to potentially review? And it looks like maybe somebody has forgotten to add the consent, or potentially they have deconsented. So this allows me to have sort of that trigger to go in and investigate what might need to be reviewed or cleaned up as well. On that review notion, I also have a very nice dashboard that allows me to see all of the data that still has sort of that outstanding review within the system. And so this CDMS, again, we have kind of our participants and then the number of records for each of these tables that we've integrated into the CDMS, but then we also have this visit notion. So again, a good CDMS will sort of take a visit or a time point notion and provide value for you as you're generating reports. And so I can see for my enrollment, but then also for some adverse events, that there is some data that is not yet reviewed. So I can click on that and go view that and make changes and modifications as I need to. I'm able to see the overall study progress as well. So instead of just looking at the data that needs to be reviewed, I can look at all of the data, or potentially all of the data that has passed review. This will allow me to understand if there's missing data. So in this case, you know, I'm really expecting a blood sample from each participant at every visit. And so understanding that there's 12 participants, I'm like, okay, there's some missing data here that we need to reconcile, clean up, maybe talk to the lab who is storing the samples or generating those samples and figure out what's happened. If I want to create common reports, there's lots of different ways within a CDMS to sort of generate these, and that's kind of what I'll show you here. But I think sort of one of the most important reports that you can generate is one around adverse events. And so here I'm going to go in and actually look at data from an individual participant, because I know this participant has undergone an adverse event. I want to make sure that we're collating that report appropriately for our safety boards. I actually get a great view of everything that we have collected about this participant 105. So I can see sort of the demographic information, any enrollment, maybe sort of lab results. But I also have this adverse event table. And so I'm able to look at and export data as I want to. Maybe perhaps I also want to insert a new adverse event before my report. Now, this may not happen necessarily in the CDMS, but this is certainly possible if your systems are still on paper or things like that. So I can fill out this information with sort of the appropriate date. I can have sort of a severity score. So here I'm going to say that, you know, somebody has a fever, and I'm actually going to sort of traverse our ontology tree to get the exact code that we need to associate with this adverse event. And so I'm not a CRA or a clinician. I don't have those codes memorized. Perhaps some of you do, in which case you could just sort of type in the code and we would make that mapping for you. So here I can also look at that path of that code even more and sort of traverse that tree and understand it as I need to. But then I have sort of submitted this additional information. My report is now ready. I actually have sort of an adverse event report that I've collated to pull in other information from some of these other tables into my adverse event report. So it might be important to understand, you know, what was the viral load at that moment? What are the lab results that we had for that participant? So I'm able to sort of export and sign the data. Here, when I'm signing, I'm able to say why I'm doing this. You know, exported for we'll call it my SAE report. And now I have sort of a snapshot of that signed data over here in my analyses. So this is another sort of great benefit that I'm able to capture information that I might be sending out to collaborators that may not have direct access to the system. Now, in most cases, ideally everybody would have access, and of course, again, the appropriate controls, but there might be times that I might be sharing this, and this gives me a full snapshot of that raw data file that I sent, so I'm able to always trace back any analyses or reports that people have generated outside of the system as well. If I wanted to create sort of a standard report for the Data Safety Monitoring Board, I would be able to do that. This is sort of another grid view of things. I can filter and sort here. So if I'm really reporting, only things from the last 5 visits or something like that, I would be able to filter that and export that information out as needed as well. A good CDMS will also have ways to analyze or visualize the data within the system. So this is a really great trend of our CD4 counts over the treatment regimens. We're able to also filter these again by our cohorts or the HIV status of our participants. We have sort of a lymphocyte cell count as well. So if I'm interested in sort of using this data, sharing it with folks, I can also export as kind of an image or a PDF, or just simply direct them to this link within the CDMS to view the data live. And then lastly, we have kind of those signed snapshots, but then also more advanced reporting needs. So maybe I've presented something or shown something around kind of this relationship between our CD4 count and total lymphocyte count in both of our sort of HIV negative and positive groups. This allows me to come back to that and understand how that analysis was created and with what data. And finally, the other thing that that I want to show here is sort of the ability to cross over to these other tables. So this is sort of a mega table that I created without code, just pointing and clicking within the application to pull in all of the vital information across each of the tables within a single table. So this could really help for that predictive analysis. It could help as I'm trying to understand trends or pieces of information about maybe adverse events within the data. But this is really a great way to generate that information as a standard report that everybody has access to. So I think for now we'll kind of stop the demo and move on to if any folks have questions. I'm happy to kind of walk through things or talk about specific reports or challenges that you might be having. I will say sort of oftentimes what folks ask is, how did all of this happen? How did all of this data come in? It's really great to show sort of the end result, but what does that process look like? And in many cases, this is what account managers are for. So what we do at LabKey is sort of walk you through this process, but most of this sort of pulling and collating of data in is programmatic. So we already have built out integrations with things like REDCap or metadata to pull in EDC data automatic on a nightly basis. We have ways to pull in laboratory data. So if they have their own system or if they want to use the CDMS as a platform to register and clean that data, there's a mechanism to do that. We also have a sample management system that is in this CDMS as well that would allow a biorepository to utilize and or again programmatically pull that data in as whatever sort of your organization sees fit. And really then from that point, once you have the data in those nice clean tables that we looked at, it becomes very easy to do the data cleaning and the validation and the verification, as well as that downstream reporting that folks are looking for. And then another question I see here is, this is great for one study, but what happens when there are multiple studies? And the reality there is a CDMS, a good CDMS will be able to apply kind of that same framework, that same notion across studies and for individual studies. And then certainly you can manage sites and that data at a larger scale within sort of the same, again, centralized system. So if you're interested in what we talked about today, if you're sort of struggling with this, or if you want a more tailored demo, personalized demo, I'd be very happy to chat with you. So please feel free to kind of schedule a demo. Yeah, and one more question here, kind of talking about reproducibility, about what are the reports, versioning, so making sure that you can have like that data in our report in different time points. And I think what this user is trying to say is how can we snapshot reports to make sure that we know, when that happened? And there are a few mechanisms within our CDMS framework that allow you to either create a live report. So some of those reports that I was showing you on the CD4 count or lymphocyte count were live. And so as the data gets updated within the system, that report gets updated. But there's also mechanisms to create snapshotted reports and actually date those and indicate that those are sort of final. And that allows then for you to snapshot a report and say, this is what I shared at a conference last week or last year. And that's really then kind of snapshotted in time. Any other questions? Okay. Well, again, we love to talk to people about their data problems. So if you're struggling and have challenges, we'd love to discuss and talk with you and see how LabKey can help. Thank you guys so much.